All right, so get a consensus. We'll get, and the first person I call gets to pick, start or end. All right, so let's see. Drew Rishardi. Drew, are you here? Drew, so tell me, what do you want to pick, the start or the end? So tell me about the start then. Is there any restriction on it? It will always be methionine. Absolutely. Drew's got it right on the money. Because the start codon encodes methionine, every single protein is born with methionine at its end terminus, as you asked. That's exactly right. Because a start codon happens to encode an amino acid, every protein starts with meth. That's the truth. All right, that's the story. All right, so therefore, all proteins start with methionine at their end terminus. Okay? All right, so what about the C terminus? Let me pick somebody. Sophia. Sophia, are you here? Sophia? Oh, there you are. Hi, Sophia, sorry. So, can you say anything about the C terminus, the end of the protein? No. I see you said no very quietly. That's absolutely right. It can be any amino acid. Because basically you roll along, you start with a start codon, methionine, alanine, leucine, blah, 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 blah. You put the last amino acid in. It can be anything. And then you put a stop codon. And then it stops. So she's absolutely right. You can have any amino acid you like at the C terminus. So, thank you. So, and can end with any amino acid at the C terminus. Okay. That's kind of big, so I want to make sure there are no questions about that. But it's a direct consequence of the way the code works. Okay. I want to draw a little cartoon of this process, and then I want to do an example, because that's the best way to really make sure that you understand what's going on. So here we go. So an example. Right? A simple gene. Okay? So the gene itself is, as always, DNA. And there's a 5' end, and a 3' end, and a 5' end, and a 3' end. All right? And you're going to have, at one end, promoter. Talked about it last time. That says start making RNA. And at the far end, you're going to have a terminator. Okay. And then what's going to happen is transcription is going to make your mRNA, which extends from just after the promoter to just before the terminator. 5', 3'. This is the mRNA, as it would have been shown in the GeneX. And that also has, somewhere along here, a start codon, which is not the same sequence as a promoter, and a stop codon down here, which is not the same sequence as a terminator. And then the next step, I'm out of cool colors, I guess I'll just use blue. Bless you. Translation makes a protein. And it's shorter because it was one amino acid for every 3 nucleotides in the RNA. So that's your basic sort of cartoon of how the gene does its thing. A couple things to bear in mind, which are sort of counterintuitive at first. Remember, we're not transcribing all the DNA, just the gene we care about. So there's DNA on either end that we are not transcribing. We are not even translating all the RNA. There are typically leaders and trailer regions with a little bit of RNA that's not transcribed. I mean, why not have your start codon right at the beginning? And why not have your stop codon right at the end? The answer is, from the point of view of Bio 111, there's no explanation for that. In real life, the sequences before the start and after the stop are involved in regulation, controlling when this gene is expressed and in what cells and in what parts of the cell. So it's actually, in real life, it's important, even though it's not obvious at where we're talking about. Questions about that before we do an example? So if you turn to the next page of today's information packed handout, it will look something like this. So putting it together. Right? So you got a DNA, you got a promoter, you got a start of the RNA, blah, blah, blah. First question, talk to your neighbor. Given that information, are you going to make RNA A or RNA B? We'll talk protein in a second. This is sort of a review from last time. Which RNA are you going to make and why? Why can you only make one? The same drill. So think about it, talk to your neighbor, or call names. If you make an RNA A or RNA B.